Hello, I am Steve Pettit. I'm the Director of Cell Culture Development at Invitria. We at Invitria develop, manufacture, and market high-performance cell culture products. In addition, all of our products are well-defined and animal-free. Today I'm going to introduce you to Zap-CHO. Zap-CHO is a new media supplement designed to maximize CHO cell culture performance. We developed Zap-CHO after talking to many scientists who had problems with their CHO cell line. For example, the cell line development scientists who struggles to get their cells to grow fast enough, so they are forced to use FBS. Or those working in seed tray expansion who would like much faster doubling times in order to speed up their timelines. Or the scientist who is getting variable results in their production because of the lot-to-lot variability of hydrolysates. We also talked to people who needed to optimize their lines for bioproduction. Another example is the process engineer who is trying to achieve higher product recovery and purity during downstream purification. With the goal of solving problems like these, we developed Zap-CHO. Zap-CHO is a robust, animal-free, defined media component that helps maximize cell growth and productivity. Zap-CHO is actually a combination of defined recombinant proteins designed to maximize CHO growth and productivity. Zap-CHO contains well-defined proteins and is consistent from lot to lot. Zap-CHO is also cost-effective, and Zap-CHO provides the performance of FBS without the animal source material. In addition, Zap-CHO is not a hydrolysate or nutrient feed. Actually, Zap-CHO represents a new class of media supplements. One of the benefits of Zap-CHO is that, as a supplement, it is effective across many media types. What you can see here is that we compared Zap-CHO across a variety of commercial media, and these are represented in this figure by the letters A through F. The effect of Zap-CHO in these 6 media were compared for the production of antibody, and that is shown on the left axis. The unsupplemented media is shown in the red color and is set to a relative value of 100. The blue bars are the same media, supplemented with 2 grams per liter of Zap-CHO. Zap-CHO increased the average productivity of these media roughly 100 to 200%. In certain instances, such as media E, in which the cell line did not grow very well, we were able to improve productivity 7-fold. The 6 media formulation comprised a diverse group of media. In this collection of 6 media, we have serum-free, protein-free, and chemically defined media. The amount of productivity increase shown by the blue bars induced by Zap-CHO was compared to 10% FBS shown by the black bars. As you can see, in every media formulation examined, the amount of productivity induced by Zap-CHO was equal or greater to that seen with the 10% FBS control. In this figure, we show the 14-day growth curve of CHO-K1 expressing a humanized monoclonal antibody. The run length was 14 days. On the left axis, we have the viable cells per mil. And what you can see here is that unsupplemented media, represented by the red line, produced at maximum cell density around day 6 about 3.5 million cells per mil. Cells grew and then reached zero viability on day 10. When we supplemented this media with 2 grams per liter of Zap-CHO, we saw the growth curve that is represented by the blue line. And as you can see here, that Zap-CHO improved the growth kinetics of this CHO-K1 line across all 3 points of the growth curve. In the growth phase, we see a greater slope, which indicated reduced doubling times. At the top of the peak, there is a greater peak cell density, in which the maximum cell density increased from 3.5 million up to 6.5 million. On the decline phase, we see increased cell viability, an extension of viability and the life of the culture of roughly 4 days. In this experiment, we compared the growth kinetics to that of 10% FBS, and that is shown by the black line. And as you can see here, the performance of Zap-CHO at this level of supplementation actually exceeded that of 10% FBS. In this study, we calculated the average increase of growth and productivity of the CHO-K1 line in 6 different media formulations. Our parameters of measurement are represented in the left-hand column. These are growth over the first 5 days, otherwise known as log phase growth, cumulative cell density over the entire 14 days of culture, and antibody productivity at the end of batch. The baseline measurements of these three parameters were set to a value of 100 in media in which no additive had been added. That is shown in the second column. We then took each of these 6 media formulations, supplemented them with 2 grams of Zap-CHO, and measured these 3 parameters. That is shown in the third column. As you can see that over 5 days of growth, the amount of viable cells increased from a relative value of 100 to 269. The 14-day cumulative cell density increased from a value of 100 to 290. The end-of-batch productivity increased from a value of 100 to 279. We compared these results to that of 10% FBS, and that is shown in the right-hand column. And as you can see, the numbers are very similar, roughly a 2-fold increase in antibody productivity. In addition, we have results that is not shown here in which we compared the effect of hydrolysates and IGF-1 to that of Zap-CHO. And we have data that supports that Zap-CHO outperforms both hydrolysates and IGF-1. One of the questions often asked is whether the proteins found in Zap-CHO can be removed by downstream processing. In this figure, you will see that all the proteins found in Zap-CHO are easily removed by protein A chromatography. We loaded cell culture supernatant that contained excreted monoclonal antibody that had been produced in a CHO cell line grown in media supplemented with Zap-CHO. The figure below shows the purity of the antibody after protein A chromatography. And what you can see is that the components of the heavy chain and light chain of the antibody are easily visible. And what you can see in the gel is that there are no traces of remaining Zap-CHO. There are many advantages to using Zap-CHO. In addition, Zap-CHO can be used in many parts of cell cloning and in different applications. The advantages of using Zap-CHO is that Zap-CHO results in faster cell growth, higher peak cell density. Zap-CHO reduces apoptosis and increases cell viability. As you've seen, Zap-CHO maximizes productivity, and it is easily removed by protein A chromatography during downstream purification. Applications of Zap-CHO in your lab include clone selection, seed train expansion, adaptation of cells to different media, and in maximizing bioproduction and an antibody titer. We would like to thank you for viewing today's presentation about in vitro Zap-CHO. If you would like a poster containing much of the data you've seen today, please contact us.